Sorry, I have to cut the recording because somebody called in my office, and so I'm just going to continue. I'm just going to what I was talking before. Yes, I said enzymes are really large molecules. As I said, they range from 10 to the power of 4 to 10 to the power of 9 grams per mole. Yeah? So they have a very high molecular weight. I cannot draw them here, yeah, but just imagine they are large molecules connected by bonds, and and it contains something like, maybe let me change the color here, something like here, you see. So this part, so this is like a space. We can call this space as active space, so that can recognize your reactant molecules. So if this is your reactant molecule, so they can recognize. So this type of active spaces, they can recognize your reactive molecules, or they might contain some active sites. Say this is site 1, site 2, site 3, and just now I explained this Oxton hypothesis. So they can recognize so three different sites from a reactive molecule. So then they can target only that particular type of reactants. So enzymes are large molecules, and it contains a lot of active spaces and may contain lot of active sites, and most of these active sites are located on the surface of your enzyme, including these active spaces. So these are the things you have to think and imagine, because you are not going to study anything at molecular level for this module, but just imagine enzymes are molecules. Obviously molecules are composed by atoms, and it has some beautiful molecular structure, and they have some active spaces. That spaces can fit your reactant molecule. It cannot fit any reactant molecule, but only a specific reactant molecule. And just now I explained one hypothesis. Just imagine your enzyme surface contains 3 active sites, and those active sites can recognize only three specific type of atoms from your reactants, which mean they can recognize only one type of reactant. So these are the things you must remember. So I think that finish our first hypothesis of Oxton. Some people, they call this as a 3-site hypothesis. So I think we can go to the next one. And another hypothesis is very famous one. It's proposed by Emily Fisher in 1890. All these hypotheses are really old, so for some reason no one come up with new ideas to explain this particular property of enzymes. So this one was proposed by Fisher in 1890. It's called lock and key. It's very easy. From the name itself you can understand. Just assume you have enzyme. You call this as enzyme, and you see here there are some nice spaces in your enzyme molecule. So you will have a substrate that will exactly fit into this type of space. So this is your substrate or your reactant. So they can go and lock. So just assume this is your key and this is your lock. So they fit each other. So that's why it's called lock and key mechanism. So this is another mechanism to explain the absolute specificity. As enzyme. So what does that mean? Like, your enzyme have a specific shape due to the way these atoms are arranged to make your enzyme molecule, that allows them to fit and lock your substrate. Yeah. So if you so your enzyme have a specific shape, and your substrate have a specific shape. So they fit each other and they lock each other, and then they can form an enzyme substrate complex. See, this part is your enzyme, and say this part is your substrate, and now we can say this as enzyme substrate complex. They just come together and then they form a complex. So we call them as enzyme substrate complex. And the third mechanism, there is another mechanism. It's proposed very recently by Koshland. To understand this mechanism, you have to have some imagination to think enzymes at some atomic level. Yeah? So just assume this is your enzyme and say this is your reactant or substrate. So what this hypothesis says is like it's almost like a key and lock and key mechanism of proposed by Fisher. They come together, but once they come together, some minute change can be observed. So once your substrate come together with your enzyme, the shape of substrate will slightly change and the shape of enzyme will slightly change, so that they lock and fit to each other, making an enzyme substrate complex. So what Fisher's lock and key mechanism says, like these two molecules, enzymes are very rigid, substrate are very rigid, so they cannot bend or they cannot dislocate from their position, or their molecular structure remains intact, yeah, like a piece of wood, so that they cannot bend. But what Koshland says is like both are slightly flexible. So once they come together, they adjust to each other, and eventually both of them they slightly change their structure. Say like your substrate can manage to squeeze inside that particular lock, and once your substrate comes together, just say this enzyme, they just hug the substrate molecule, so making a enzyme substrate complex. So, and this is a more reliable model. And a proven model. Many computational biologists, they tried to test this hypothesis, and they really observed using molecular level simulations, and they observed once the substrate goes into the enzyme packet or the active packets, both of them slightly change their structure, and then they form an enzyme substrate complex. So these three are the these are some hypotheses that you need to know if I ask you why enzymes are specific, these things you should remember. Yeah? So I will upload the notes. It's in the PowerPoint. So try to read the PowerPoint where I showed some images that can help you to look enzyme at atomic level. And one more thing, like since I talked a lot about like the structure of enzyme, you should always remember, just assume this is your enzyme structure, and this part is your active packet or active site or active packets. So enzyme can be in this particular shape only at specific or some specific range of temperature, maybe not at one temperature, some range of temperature range and pH. Yeah? So if you alter the temperature and pH, then enzyme will lose their structure, and it anymore won't. So you will not have these active spaces. So no more active space if the enzyme lost its structure. So no more active spaces. So which means this enzyme is practically useless because it cannot recognize your substrate. Imagine this is your substrate. It cannot recognize your substrate, which means this enzyme is useless, which means it won't have any catalytic properties. So this is something you should remember. And why a slight fluctuation in temperature can change your enzymes? Because enzymes are composed by atoms. Just to say, these are your atoms. Just that makes your enzyme structure, and they are connected by weak intermolecular forces. So once you increase the temperature, that's more than enough to break these intermolecular forces connecting those atoms, and eventually they will lose the shape. So these are the things you must remember. And one more thing, this is an engineering module, so I'm not going to talk a lot about protein structure. Since enzymes are proteins, you should know how protein structure looks like. So I'm going to upload an YouTube link. You must watch this link, because I'm going to ask some questions based on what's shown in this particular YouTube link in the exam. So expect some questions from this particular. It's a small video, but you should have some understanding. So if you watch this video, you will understand how enzymes will look at molecular level, and why they have some unique structure, and how the atoms are connected to each other, giving that beautiful structure to the protein, which acts as a catalyst. And that's why we call them as enzymes, and that's why enzymes are also called as catalysts, biological catalysts. So I think I covered a bit. I'm just going to summarize what we saw. So far we saw properties of enzymes in this lecture and this previous lecture. So lecture 7 we covered properties, both part 1 and part 2, which we are reading now at the moment. So enzymes or proteins, they are large molecules. They are not tiny molecules like water or carbon dioxide, and their molecular weight ranges from 10 to the power of 4 to 10 to the power of 9. And cells, you can call them as enzyme manufacturers. So you have a cell, and then you have enzymes inside. So you break the cell and take the enzyme out. So that's why we call enzyme manufacturer. And enzymes are not alive. So they are a biological product. Yeah? So you break the live cell and take the enzymes out, and then we discuss the difference between enzyme and versus a live cell. So live cell, they can multiply, both can break your substrate and convert them into product, but live cells can digest. And then I talked about what's the job of an enzyme based on a small example from the studies of from the group of I don't remember. Yeah, I showed some studies performed by Japanese scientists. Yes, yeah, they wanted to digest the polyethylene terephthalate using a particular type of bacteria. And then I said about this bacteria, what it do is like it secretes an enzyme, peptidase, that will convert your substrate into product, and a substrate into smaller compounds, and the bacteria themselves, they can consume those tiny molecules and they can digest. So that will show the key difference between the live cell and the job of an enzyme. Yeah. And then another thing, I talked about the structure of your enzyme. We talked about the molecular structure. I said they have some active sites and they have some active spots that can fit your substrate. And then I talked about specificity, a very important property of enzyme. I talked about absolute specificity. I talked about group specificity or group specific, and then to explain them, we talked 3 different hypotheses. One is proposed by Emily Fisher, another one by Coastland, another one by Oxton. So Oh, one thing I forgot to mention, Coastland model. Sometimes they call it as induced fit model. I showed you this one, yeah, like here. So this particular model. So I said once the substrate goes inside, they try to adjust themselves and then they lock with each other. So it's the substrate, once it goes inside, it induce some action on the enzyme, and that's why enzyme, they'll try to change their shape a bit and try to lock them completely. So that's sometimes they called as induced fit model. Instead of model, you can call as a hypothesis, because these are scientific hypotheses, yeah? They just imagine why enzymes are specific, and they came up with this type of hypothesis. If you have a better imagination, you can propose your new hypothesis and someone can test it at a later stage. And then I talked about initial velocity. You must remember what is initial velocity, yeah? Because this is really important in enzyme kinetics, yeah. Many people try to derive Michaelis Menten kinetics, and then they even solve 100 different problems without knowing the meaning of initial velocity. So initial velocity is a very is a very important parameter. Like if you were doing some experiments in the lab, you can get some information about how much products are formed with respect to time. Like I said, you can monitor your product concentration and then you can plot them, and this is your initial velocity. So change in product concentration with respect to your time in this initial region, where the the change in product concentration with respect to time is linear, yeah? So that's why we call them as initial velocity. And the unit is, if product is expressed in milligrams per liter, time is expressed in minutes, unit is milligrams per liters per minute. So that's the meaning of initial velocity. And initial velocity, it also means activity. Activity of your catalyst. And if you know your initial velocity and if you divide it by your total mass of your enzyme, then that will be called as specific activity. So these are the things you should remember. And then I talked about turnover number. So turnover number, it's something related to catalytic activity. But here everything is expressed in terms of numbers instead of moles, like grams of products formed per unit time. We won't say that. We will say number of molecules of products formed per unit time. So that's how you define turnover number. And then I talked about stability. I said enzymes have some specific shape. If you change high temperature, then it will lose its shape. So it will transform. So it won't be, so it won't, how to call it, will become useless, yeah, once it loses its structure. So it won't have those nice active spots, and your active sites will be distributed randomly, which means they cannot recognize your reactants, so which means it won't do the job, yeah. So if enzyme needs to convert your reactants into substrate, it must form that enzyme substrate complex. And why is that? That's what we are going to see now, yeah. I'm just checking the notes whether we are going in the same order or not. Okay, coming back to this stability. I said to you, like, imagine this is your enzyme, and you have 3 active sites here, or you can call this is your enzyme and this is your active space here, and just assume this is your substrate which 3 active sites. So say this is your substrate with 3 active sites. They can recognize, this site can recognize this one, this site will recognize this, and this site will recognize your third site. Or if the shape of your substrate fits exactly to the active space, then they can come together using this Fisher lock and key hypothesis. This is comes from your Untitled model, and then they can form, say this is your substrate, this is your enzyme, and then they can form this enzyme substrate complex. So this is really important, and if you alter the temperature, then they will lose its structure and it won't form this enzyme substrate complex. So no enzyme substrate complex. And why do we care? Why do we care that it should form about this particular complex, enzyme substrate complex? Because your reaction, this enzymatic reaction, will not proceed forward unless we don't have this, unless we have this enzyme substrate complex. Enzyme substrate complex. In fact, enzymatic reactions are governed by the concentration of your enzyme substrate complex inside the reactor. Yeah? So we are going to see why is that. So how to model enzymatic reaction, or if you want to call, or if you want, you can say, how can you explain or what happens during an enzymatic reaction. We don't have to propose any new theory or hypothesis, because Leonard Michaelis, I don't know how to spell, and Maud Leonora Menten, they proposed some scientific or theoretical idea to explain what really happens in an enzymatic reaction. So they said, imagine you added your substrate into the reactor and you added enzyme, then you start the reaction, give some correct temperature and pH, and then you start your reaction, and then you will see the product formation with respect to time. You have time here, you have your product concentration. Your product concentration is going to increase. So what really happens with enzyme and substrate? Yeah? So this is what Michaelis and Menten thinks. So they first give some notations for enzyme. They used the notation E square bracket, that means concentration of your enzyme. And then they call S as concentration of substrate. Then they give some notations for enzyme substrate complex, concentration, enzyme substrate complex. Whatever we saw so far will make sense when we are going to derive this equation. And then he gave P for product concentration. And so this is what, so they begin by giving some notations. No matter what enzyme you use, no matter what substrate you are going to deal with, just assume you know the concentration of enzyme, you know the concentration of your substrate, and just assume they come together, they make some enzyme substrate complex, and eventually you are going to monitor how much product you are going to get in that reactor. And then they go on to make few assumptions. They said, like enzyme plus substrate, it won't directly give products. So this is no way, sorry. So what they said is like you get product in an enzymatic reaction and via two elementary reactions. Yeah. So this is what they said. Enzyme plus substrate, they tend to form a complex called enzyme substrate complex, and from enzyme substrate complex it gives to enzyme and then product. So this is one reaction, or you can call reaction 1. You can call this as reaction 2. In fact, this is not even a big chemical reaction. This is just how do you call? We can call it like a physical attachment of both enzyme and substrate, or if you want, you can call enzyme and substrate. They associate together to form an enzyme substrate complex. There is no harm in saying this as a reaction, because at the end of the day you are getting something new. This can be an intermediate phase, but still you can call it as an intermediate phase, or just call it as enzyme substrate complex as called by Michaelis and Menten. So this is, and this is the first assumption they made. So enzyme and substrate, they associate together to form a new complex called enzyme substrate complex. And this enzyme substrate complex, once your enzyme, and say this is your substrate, they come together to form enzyme substrate complex, then enzyme do the job, which is to convert your substrate into products. So what it do is like it convert your enzyme, and it will convert into new product. So this is your product, leaving the enzyme free. So enzyme really doesn't participate in your reaction. It only catalyzes the process. It takes your substrate and then convert it into products. But there is some intermediate process here. So you have two steps. So this is the first assumption. You have two elementary steps. So one is the formation of an enzyme substrate complex, and followed by this breakdown of an enzyme substrate complex into enzyme plus products. So this is your first assumption. So the second assumption is that this enzyme plus substrate, they said this is instantaneous. So this is instantaneous. So once you take your reactor, you add your substrate and you add your enzyme, and they said immediately they trying to associate together, and then they form this enzyme substrate complex. So the time scale involved is almost negligible. So it's spontaneous. So once an enzyme see the substrate, they associate together and then they form an enzyme substrate complex. So this is purely physical. So they come together and then they make this complex. So that's it. So, and another thing is this process is reversible. So it's not there is no strong bond between enzyme and substrate. So this process is completely reversible. So that's the third assumption made by Michaelis and Menten. So the formation of enzyme substrate complex, the process of formation is reversible, associated with a kinetic constant Km. Go back to here. Let's use new color Km. So this constant is related to the formation of enzyme substrate complex. Yeah? So if Km is faster, high, which means enzyme substrate complex are formed more rapid. Yeah? So higher this number, which means the formation of enzyme substrate complex is very fast. So far we saw 3 assumptions, and then the 4th assumption. Yeah? I'm going to write again. This is, we have 2 arrows, which mean reversible, and Km. That's the kinetic constant associated with the first process. And then this enzyme substrate complex. This is the catalytic process. So you want enzyme and substrate, they come together, and then enzyme starts its job, which is to catalyze the process. And it converts your enzyme to products, leaving the substrate enzyme free. So this will do the next process. It will take the new substrate molecule and then convert your substrate into products. So this is the actual catalytic chemical reaction is happening. So we call Kcat. And this is an irreversible process. Once your substrate are converted into products, you cannot convert your products back to enzymes. Yeah, and that is another reason for this. This assumption is not bad assumption. You see this, you have an enzyme, and if this is your product, they can recognize, sorry, this is your substrate, this is your enzyme. They can recognize only your substrate. If your product is a different shape, they cannot your enzyme cannot act or cannot associate with your product. Yeah? So due to the perfect this molecular recognition capability, or if you want, you can call the specificity of your enzyme. So the enzyme can only specifically attack your substrate, not your product. So that's why this is an irreversible process and associated with a kinetic constant Kcat. Yeah? And another 5th assumption is like the first step is fast. This is a reasonable assumptions made by Michaelis and Menten, because this is purely a physical process. Yeah, there is no bond breakage or a new bond formation is happening. They just come together and then they attach together like an adsorption process. So maybe just due to some physical force, your enzyme and your substrate. So it's combination of physical force and the specificity of your enzyme. Your enzyme take your substrate, they come together, they form a nice enzyme substrate complex, and therefore you can call this is really a fast process. Yeah, so it takes few minutes, or forget the minutes. The time scale is relatively faster. So just they can recognize each other, they can form an enzyme substrate complex. But the second step, which is the actual chemical reaction, is happening where some molecules are broken into new molecules, which we call them as products. So the second step is the slowest step. Second step is the slowest step, and that's why we are going to call this as a rate determining step. I said to you, I showed you some example before as well. Just imagine if some guy is going in a car very fast, say 100 kilometers per hour, and imagine there is some other guy in a bicycle going at a speed of 5 kilometers per hour. This guy have to slow down and wait. He have to stop, yeah, and then he have to match with the speed of this guy who's going in the bicycle. So this guy is like a slow reaction, and this guy is like a fast reaction. So at the end of the day, he decides the time. So and chemical reaction works in the same way. Imagine like if once you take your substrate, you take your enzyme, you add together, and everything comes together in 30 seconds to make enzyme substrate complex. And if it takes 1 hour to break your enzyme and substrate complex to products plus your enzyme, you have to wait for 1 hour. So this is the slowest step, but the rate determining step. Yeah? Because at the end of the day, your objective is to form products, not enzyme substrate complex. Yeah? So this is a reasonable assumption. And since the second step is also a chemical reaction, normally it takes some time to break your molecule from its own state or its original state to new molecules, which we, as I said, these new molecules are nothing but your products. So this is another assumption. The first step is purely physical and very fast process, and the second step is the slowest step and thus the rate determining step. So these are the assumptions made by Michaelis and Menten. And then now we can give a mathematical treatment for this reaction. Yeah? Let's start writing this equation. I hope you all know what is an equilibrium reaction. You must have studied this in the first year general chemistry, I hope. So this is an equilibrium reaction. So normally we write kinetic constant Km, concentration of your reactants, divided by concentration of your products. And so this is your rate controlling step. So your whole reaction rate is controlled by this particular step. So your rate is equal to K catalyst. Or if you want, you can say your rate is proportional to the concentration of your enzyme substrate complex. So this is the sixth assumptions made by Michaelis Menten. What he said, your rate is controlled by the slowest reaction step, which is normal, and that rate is proportional to the enzyme substrate complex here. So rate of an enzymatic reaction is proportional to the concentration of enzyme substrate complex. And now we started the enzymatic reaction with the substrate and the enzyme, but your reaction can proceed. Your rate of reaction will be determined only after the formation of enzyme substrate complex inside the reactor. So that's the main key assumption made by Michaelis and Menten. So if you want, if you want to remove the proportionality constant, you can add a constant. If you want to remove the proportionality factor, so you need to add a constant. That constant will be equal to your rate kinetic constant, which I use the notation Kcat. Rate constant for the enzymatic reaction. So far so good. So what is this rate? Rate means, as I said, you start a reaction and then you're going, so this is your enzymatic reactor here, this one. You just imagine you have some probe to monitor your product concentration, and then you plot the P versus time, grams per liter. Your product concentration is going to increase. So this rate R0 is nothing but this one. Change in product concentration with respect to time. This is nothing but initial velocity. Yeah, so if you want, you can write like this: R0 is equal to change in product concentration with respect to time is equal to Kcat into enzyme substrate complex. Yeah? I hope you all can follow. Yeah? If I see you, I can stop and ask you whether whether you can follow this derivation or not. This is a really important derivation, yeah, in bioprocess engineering. You cannot leave this subject without learning the derivation of Michaelis Menten expression. This is such an important expression, and it's not only to help you to understand what's happening in an enzymatic reaction, it will also help to deal with other type of problem that you might encounter later. Like imagine when there is some inhibition effect happening, your enzyme won't have that particular property to catalyze because you have some impurity or some foreign compound, we call it as inhibitor. Then you need to know what's actually happening inside the reactor. And even for such case scenarios, you need to know what's the assumption of Michaelis Menten and what's meant by enzyme substrate complex, and why your reaction is proceeding in the forward direction, and why enzyme substrate complex plays like decides the rate of your chemical reaction. So it's really a must. So that's why I put some extra effort in deriving this equation. Yeah? And also, when you need to solve the problem, you cannot solve the problem unless you know the theoretical assumptions behind the Michaelis Menten expression. Yeah? And in the exam, you might expect some questions based on what you are learning at the moment, especially from the you might need to know what is an initial velocity, how to get it from a graph, and how can you use that to calculate some of the kinetic constants involved in the enzymatic reaction. Okay, let's go on to the next step. So I said, just imagine like you take your reactor, you added enzyme, you added substrate, it formed enzyme substrate complex. So at any time t, just assume your reaction started already, which means you might already have some freestanding enzymes, and you might have enzyme substrate complex inside your reactor. So your total enzyme concentration should be equal to E total should be equal to concentration of enzyme, which is freestanding inside in the solution in the reactor, and also enzyme which are already associated with substrate in the form of enzyme substrate complex. Yeah, so this will be your total enzyme concentration at any time. So concentration, which is your freestanding enzyme, so these are ready to react with new substrate, and these are enzymes which are already attached or associated with the substrate in the form. So if you want to know the concentration of enzyme at freestanding enzyme at any instant of time, so you can write that will be equal to E total minus concentration of your enzyme substrate complex. Yeah? I think it's better we give some number to all the equations. So go back here. Here I derived the equilibrium constant. Let's give this as equation 1. And here I showed what is your reaction rate. So this we can call it as equation 2. And this one we can call this as equation 3. And this one we can call them as equation 4. So what we are going to do is substitute equation 4 in equation 1. So we know Km is equal to so you substitute this value here. So you will get E total minus enzyme substrate complex plus ES divided by E tot minus ES, yeah. total minus you just expand this equation, so this will be equal to E total multiplied by your substrate concentration minus enzyme substrate concentration minus multiplied by substrate concentration. Yeah, you can just put the square bracket all around E total. So this square bracket means concentration, just to maintain the consistency. So divided by your enzyme substrate complex, concentration of your enzyme substrate complex. So this is your Km value, and then you can just take this value here equal to E total multiplied by your substrate concentration minus enzyme substrate concentration, and then you can take all this value on one side, your enzyme substrate complex, say like you'll get something E total multiplied by your substrate concentration. So from this we can calculate concentration of your enzyme substrate complex. So why are we doing this? Because we cannot measure, once you started your reaction, you don't know what's the concentration of your enzyme substrate concentration. So that's why we are trying to find a relationship. We are trying to find what's the value of this enzyme substrate complex will be. So this will be equal to E total multiplied by your substrate concentration divided by your equilibrium constant plus your substrate concentration. So just call this as equation 5. Say this is equation 5. And what we are doing so far, we are trying to calculate the rate of the reaction. Yeah? So change in product concentration is equal to K catalyst multiplied by your enzyme substrate complex. Yeah? So we need to substitute this value here. So we know enzymatic reaction, the rate of the enzymatic reaction depends only on the concentration of enzyme substrate complex. Obviously we cannot measure them inside the reactor, but we can connect that concentration with some kinetic parameters, which is like your equilibrium constant and then your total enzyme concentration. We know this one because we know how much we added in the reactor and we know how much substrate concentration we have. So this is really easy. So you substitute your enzyme substrate concentration here. So you will get R0 is equal to K cat E total multiplied by your substrate concentration. You divide it by Km, which is your kinetic, another constant or equilibrium constant, multiply plus your substrate concentration. Yeah? So this is the derivation proposed by Michaelis and Menten, or if you want you can call dP by dt is equal to this one. Yeah? And in many books, many textbooks, you will see a different format. Yeah? So they instead of R0, they use another parameter. They call it as mu. This is nothing but activity of your catalyst. Yeah? So this is something like I said the unit is milligrams per liter per minute is equal to K cat and E total enzyme total multiplied by your substrate concentration divided by Km plus your substrate concentration. So let's call this as equation 6. Let's call this as equation 7. 6 and 7 are same, but we use a different notation for rate of this reaction, mu. If you want, you can call this R0 is equal to mu, and you can call this as reaction rate, or you can call this as initial velocity, or you can call this as activity of or catalytic or activity of your catalyst. So all mean the same, yeah? And then you can simplify this equation 7. So you can rewrite equation 7. So this K catalyst multiplied by total enzyme concentration, that's something we call it as mu max. So just rewrite that equation. So mu max multiplied by your substrate concentration, Km, plus your substrate concentration. So this is something called maximum catalytic activity of enzyme. So one thing you should remember, if I give you a catalyst and if I ask you to characterize the catalytic activity, that always corresponds to this value mu max, yeah? So mu max and catalytic activity, sorry, yes, yeah. So mu max refers to the maximum catalytic activity. I will come back to this one. What does this mean exactly? So how do you call, like mu max is a cat. How do you, maybe I will say it quickly so that way you will understand. Say you have time versus product concentration here. Imagine you started your experiment with some enzyme concentration of 1 grams per liter, another with 2 grams per liter, another with 4 grams per liter, another with 5 grams per liter, and another with 6 grams per liter. So you can calculate initial velocity from the initial linear slope, and then if you plot your substrate concentration here, say 1, 2, 3, 4, 5, and then 6 grams per liter, and then you plot your initial velocity, or if you can call it as mu, you will get something like that. So after 5 and then 6, maybe I put it in the next page, it's so confusing. Come back to this graph and you do another experiment at 7 grams per liter, they produce the same result, 7 grams per liter or 8 grams per liter. So what it says is, like, at some point it reached a maximum activity, you know? I think I explained very wrong. Please ignore this. It's not the enzyme mass. Just imagine you started your experiment and then you're monitoring your product concentration, grams per liter. You started your experiment with 1 grams per liter of substrate. So that's your initial substrate concentration. 2 grams per liter, 3 grams per liter, 4 grams per liter. And then if you do another experiment at 5, it produces same result. Another experiment you produce at start from 6 gram per liter, you get the same result. And now you take the initial velocity here, dp by dt, that's your initial velocity, and then you plot it against your substrate concentration. You will get, say, 1, 2, 3, 4. You will get something like that. 1, 2, 3, 4, 5. It reaches a constant, yeah? So this value is called mu max. So this is the maximum catalytic activity of that enzyme. So if somebody asks, okay, this is the catalyst I'm going to give you, and tell the property of the catalyst. So you define that the property of the catalyst by performing different experiment at different substrate concentration until you reach this value of mu max. So mu max is the property of enzyme. is property of an is a material property, and that material is your enzyme. Yeah, so and this mu max is related to catalytic, your rate constant and the total enzyme concentration. So this is really important. Yeah, so whenever I talk about catalytic activity, catalytic activity, you should remember you can define a catalytic activity only if your reaction is happening at the maximum limit, or if you want you can say like you can define the catalytic activity of your enzyme only if your catalysts are converting your substrate into product at the maximum rate, which means your initial velocity should be at the maximum. So that's only then you can calculate the catalytic property of your enzyme. So hope you are following this one. Yeah. So this part is fine. This is very tricky, but you should pay attention. You cannot define a catalytic activity unless you know that your initial velocity should be at the maximum. So I hope you understand so far, and just don't remember mu or 0 all refers to same, or you can call this as initial velocity, or you can call this as a reaction rate. So and if you want you can call this as a catalytic activity. And then if you want you can say this mu value is equal to mu max into substrate divided by Km plus your substrate concentration, and this final expression we call it as Michaelis Menten expression. So this mu value have the unit of milligrams per liter. This mu max will have the unit of milligrams per liter. Your substrate concentration will have the unit of it depends on how you express your substrate concentration, milligrams per liter. Sorry, this one will have unit of milligrams per liter per minute, milligrams per liter per minute, because this is P versus time. So this is changing P versus changing time, milligrams per liter by time, which is in minutes. So that's the unit of your mu value and mu max. And the substrate concentration have a unit of milligrams per liter. And Km, it has the unit of substrate, so thus the unit is milligrams per liter. So always try to pay attention to the units. Yeah? So why I talked about the units? I hope you all remember I showed you in the previous lecture 6 or 5 monod kinetics that have the same expression, where we called this as a monod expression and we call this as monod kinetic constant. In that one your mu value have a completely different unit, which is some numbers per minute. So this number refers to number of cell replications happening per unit time. Yeah. So when it comes to enzymatic reaction, your mu refers to initial velocity or reaction rate, but not something related to replications, because you obviously know enzyme is not a live material, I said to you. So which means that enzymes are not going to replicate. All they can do is it can catalyze the process of converting your substrate into products. So these are the key things you should remember, and that's how you differentiate this particular parameter mu in an enzymatic reaction that has a unit of milligrams per liter per minute, or in a microbial reaction where you are going to deal with live cells. So microbial reaction, so where we have unit of numbers per minute. So this refers to number of cell replications per minute. So these are the key differences between monode expression and the Michaelis Menten expression, though it looks similar. So try to remember this one. And since I talked about unit, I can give you something else. Mu is equal to mu max substrate divided by Km plus your substrate concentration. So as I said, this is expressed in milligrams per liter. If you want, you can divide both sides by total enzyme concentration. E total substrate Km plus your substrate. So in this case, this one will become like specific activity of your enzyme. Specific activity of your enzyme. No, you cannot do that, yeah? Just strike it out. Because your specific activity of your enzyme corresponds to something like your initial velocity should be at the maximum, yeah? So forget this idea. I'm going to upload the PowerPoint which will go well with this lecture in the Sulis. There I posted one slide where you can download with some details like where to download the original paper of Michaelis and Menten. In fact, Michaelis and Menten, they published their work in German, and some professor from University of Texas, he recently translated it. I think it's published very recently. I don't remember the year, but you can go and download it. Yeah, I put the link in the PowerPoint. It's not necessary for your exam, but if you are really interested in how these people they thought about an enzymatic reaction and how they come up with this beautiful idea, and like who can imagine like enzyme and substrate form an enzyme substrate complex, and if you want a reaction to proceed in some forward direction, all you need to know is the concentration of your enzyme substrate complex. Yeah, that's a beautiful thinking, and they wrote this paper. If you are really interested, go and check that paper, and I suggest you to read that paper for another reason. Probably all of you in this class would have read many research articles for writing assignment in my module or for other modules, but this particular paper was not written in a scientific way. They wrote it very casually using some very colloquial terms. So that's why I recommend you to read this paper. It's really a beautiful paper to read. And now I'm going to do some not calculations. I'm going to give some tips how to calculate or how to get these values, this kinetic constant like Km. Let me use some other color. This Km value which is here, this Km value which is here, and this maximum initial velocity or maximum growth rate, not growth rate, maximum initial velocity, mu max. So how to get that one? To get that one, I'm going to stop recording because there is some problem with my notebook. I will stop and upload it, and then I'm going to record immediately using a new file name. Thanks for listening.